Hello students, today we are going to discuss very important questions of biology. First question is, in which of the following forms is iron absorbed by plants? Option A, free element, B, ferrous, C, ferric, and D, both ferric and ferrous. Correct answer is C, ferric. So according to NCERT, plant absorb iron mostly in the form of ferric, Fe3 positive ions. However, plants in acidic soil can absorb iron in ferrous, Fe2 positive, as well as ferric, Fe3 positive form. So it is an important constituent of proteins involved in the transfer of electrons like ferridoxin and cytochromes. So it is reversible oxidized from Fe2 plus to Fe3 plus during electron transfer. It activates catalyze enzyme. It is essential for the formation of chlorophyll. So correct answer is C. Next question is the correct order of steps in polymerase chain reaction PCR is. Correct order: A. Denaturation extension or annealing. B. Annealing extension denaturation. C. Extension denaturation annealing. And D. Denaturations annealing and extension. करेक्ट आंसर इज डी. जो पीसीआर होता है उसमें 3 बेसिक स्टेप्स होते हैं: डीनेचुरेशन, एनिलिंग एंड एक्सटेंशन. जो डीनेचुरेशन स्टेप में है वो डीएनए इज हीटेड एट हाई टेम्परेचर 94 to 96 डिग्री टू सेपरेट द टू स्ट्रैंड्स. और जो एनिलिंग में है द टू oligonucleotide primers anneal to each single stranded template DNA. And the step is carried out at lower temperature 40 degree to 60 degree Celsius. The final step is extension, wherein Taq DNA polymerase synthesize the DNA region between the primers using DNTPs, deoxynucleotide triphosphates and Mg2 positive ions. Next question is, in India, the organization responsible for assessing the safety of introducing genetically modified organisms for public use is. A option is Research Committee on Genetic Manipulation. B. Council for Scientific and Industrial Research. C. Indian Council of Medical Research. And D. General Engineering Appraisal Committee, GEAC. Correct answer is D. In India Genetic Engineering Approval Committee, GEAC, according to NCERT, is responsible for assessing the safety of introducing genetic modified organism for public use. जो GEAC है वो comes under the Ministry of Environment and Forestry, MOE, while the Review Committee on Genetic Manipulation, RCGM, comes under Department of Biotechnology. And जो Council of Scientific and Industrial Research, CSIR, is the largest research and development organization in India. Or the Indian Council of Medical Research, ICMR, is the apex body in India for the formulation, coordination and promotions of biomedical research. Next question is, use of bioresources by multinational companies and organizations without the authorizations from the concerned country and its people is called. Option A, biodegradation. B, biopiracy. C, bioinfringement. And D, bioexploitation. करेक्ट आंसर इज बायोपारसी. जो बायोपारसी हैं वो यूज़ ऑफ बायो रिसोर्सेज by multinational companies and other organizations without the proper authorizations from the countries and the people concerned without compensatory payment. और जो बायो इन्फ्रिगमेंट है वो क्या होता है? कमीशन ऑफ अ प्रोहिबिटेड एक्ट विद रिस्पेक्ट टू अ पेटेंटेड इन्वेंशंस विदाउट परमिशन फ्रॉम द पेटेंट होल्डर. और जो बायो एक्सप्लोइटेशन है वो मीन्स टेकिंग एडवांटेज ऑफ बायोलॉजिकल रिसोर्सेज ऑफ अदर कंट्री विदाउट परमिशन. And biodegradations is biological breakdown of organic material by bacteria, fungi, etc. Next question is, which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocyte? A option lambda phase, B triphased, C retrovirus, and D PBR 322. करेक्ट आंसर इज रेट्रोवायरस. जो रेट्रोवायरस होते हैं यूजली यूज्ड एज अ वेक्टर फॉर इंट्रोड्यूसिंग अ डीएनए फ्रेगमेंट इन ह्यूमन cells. They are used as vector in gene therapy to introduce the desired gene so as to replace the functioning of a defective gene. Example, SCID, जिसे Severe Combined Immune Deficiency, is caused due to defect in the gene for the enzyme adenosine deaminase. In gene therapy against it, lymphocytes are extracted from the bone marrow of the patient. These are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is the introducing into these lymphocytes. These are re-injected into the patient bone marrows. लैम्डा फेज अलाउ क्लोनिंग ऑफ डीएनए फ्रेगमेंट्स अप टू 23 kb लेंथ और जो टाइप प्लाज्मिड है यूजली यूज्ड फॉर प्लांट्स एंड PBR 322 is an artificial cloning vector usually used for bacteria.